Hi, this is Dr. Lapnow again. Now that we've learned about some basic properties of atoms and how they react and some bonding, we are going to take a look at molecules, particularly biomolecules that are found within the body, and some properties of these biomolecules. Here are the objectives for our lesson today, so take a look at these. Again, these just outline the things that you are expected to know and learn from this information. So to start with introducing biomolecules, biomolecules are organic molecules, meaning that they are what make up most living things. And biological macromolecules is another term for these biological molecules. And they're a subset of organic molecules. There are four major classes of biological molecules, and these include all of the things that are found within our body that kind of make up cell membranes, that make up organelles, that make up tissues and fibers and all those sorts of things. So inorganic molecules, on the other hand, are molecules that are polymers, metals, plastics, things like that that are not living. So the four classes of biomolecules are lipids, nucleic acids, proteins, and carbohydrates. And within our body we have all of these that kind of combine in various ways to make up all of the components of our systems, of ourselves, and of our cells. Biomolecules contain a backbone of carbon, atoms that are joined together, covalently bonded, and then also have hydrogens attached to them. And there can be different formations of these carbon backbones. They can be linear, and I have a couple examples shown for you here that are just different ways to depict molecules. And here is an example of sort of the ball and link type formation. Here's kind of a longer hand illustrating all of the carbons and hydrogens, and then here's sort of a shorthand structure of that. You don't need to necessarily know these different structures, but if you see them in readings or things like that online, you kind of understand that each of these bends, for example, here indicate a carbon, and they are typically saturated with these hydrogens that are linked to them. There's also different formations like these that are forming sort of these ring-like, these carbon rings, and they can have single bonds, double bonds. There's a whole bunch of formations. So this is the backbone of biomolecules, and they can have other things attached to them other than hydrogens. There could be nitrogen or phosphorus or sulfur, sulfates. And they can be polar, they can be nonpolar, they could have waters attached to them, acids attached to them. So there's a whole bunch of varieties of things that can be attached to them. So that's sort of the backbone of all of these biomolecules. We have these specific groups of molecules that can be attached to a carbon backbone, whether it's a ring structure, whether it's a linear structure, and these groups can have specific properties. They could have polarity, they could be an acid, they could be a base. So we call these types of groups functional groups. And the functional groups are things that actually do a function and actually determine how this biomolecule will interact with other molecules. And I have some circled here on this. Here's a carbon ring, but then there's three different functional groups here, and this will really determine how it interacts with other molecules. Most of these functional groups are polar, and they're able to bind hydrogen. Some act like acids, some act like bases. It really depends on the composition. So I have a table down here of common groups of functional groups. Carboxyl is carb and ox, so we have a carbon and oxygen in here, and that's where it gets its name. And an amino group with a nitrogen. Here's a hydroxyl group, meaning one oxygen and one hydrogen, and then here's a phosphate group. So there are several different, these are really, really common functional groups that will determine binding and interactions with other molecules. Here's a table of some of these major functional groups, what they look like, their chemical structure, where they can be found and their importance within the body, and then some examples of where these functional groups are commonly found and how they are important in those particular molecules. For example, the carboxyl group is important for amino acid binding from one amino acid binding to another amino acid, and we'll talk about that in a little bit. They're also really important in the reactivity of fatty acids. We have amino groups which are really important in amino acids, which is kind of where they get their name. Carbohydrates, fatty acids, and amino acids are also all have hydroxyl groups. Phosphate groups are important in like nucleic acids and things like that. So this is an example of the 4 major functional groups, how important they are, and then where they can be found as some examples. Biomolecules are complex molecules that are made of smaller individual components. So let's cover the basics of these. A monomer is an individual subunit that, when linked with other monomers, mono meaning one, then they form a chain of monomers, and we call that a polymer, poly meaning many. So all of these biomolecules have smaller subunits or smaller monomers that make up their larger polymers. And for example, and we'll cover each of these individually, but just to give you an overview, carbohydrates, large carbohydrates are polymers, and they're made up of smaller sugar monomers. Nucleic acids like DNA and RNA are made up of smaller monomers called nucleotides. And proteins are large chains of smaller monomers called amino acids, and all of these amino acids linked together would make a polymer. So that's the idea of monomers and polymers. In order to take monomers and bond them together with other monomers to make a polymer, this is done through a specific process where one end of one monomer is joined with another end of another monomer to make a bond between the two. And to join two monomers together, there is usually a hydroxyl group at the end of one monomer and a hydroxyl group at the other. And so in order for these to come together, we need to remove some of these hydrogen and oxygen. And so what happens is these two monomers will come together, and off will come 2 hydrogen and an oxygen, and that forms water. And then what's left is an oxygen that can then bind these 2 monomers together to make a bond. And in doing so, we create a molecule of water. So this type of joining together of monomers together is called dehydration. And to dehydrate something is to remove the water from it. So think of removing water. Or another term for this is called condensation. And think about a glass of water, for example, and it has condensation on the outside. So water has like appeared when it wasn't there before if you have a cold glass of water. So condensation or dehydration synthesis is removing a molecule of water from 2 adjoining monomers so that they can join together to form a polymer. And then a new covalent bond is formed. The opposite reaction is to take 2 already joined monomers that are probably in a polymer and to pull them apart. And in order to do this, it's just the exact opposite of what we saw with the dehydration synthesis. Take a water molecule and it sort of inserts itself in between this bond, and by inserting that oxygen into hydrogen, it sort of pulls those apart. And using water to break apart a bond is, lysis means to break and hydro is water. So hydrolysis is to use water to break apart 2 bonds. And then those will, maybe a polymer will be broken back down into its monomer, for example, or other bonds can then be formed. So we're going to start by talking about the first biomolecule, proteins, and kind of go through some of the important aspects of these proteins. Proteins are one of the most important biomolecules of our body. They make up virtually everything. We have fats and we have carbohydrates, and of course we have DNA, but pretty much everything else just has a protein backbone. So think of phospholipids. Yes, they have lipids, but there's proteins embedded in that lipid bilayer. There are proteins that are signals, like chemokines, for example. There are proteins that are receptors. There are tons of proteins that are enzymes. So there are so many functions of proteins within our body They are important in the immune system, and like antibodies, for example, are a type of protein. Chemokines, cytokines, important for helping cells to transport things throughout the body, whether it's binding to a molecule, whether it's movement of molecules and scaffolding. It could be things that are found in the extracellular matrix that are important for strength or important for flexibility. Proteins make up all of your filaments of the muscle and strength of the skin. They're important for hormones. A lot of hormones are protein-based molecules. They're important for providing storage, important for communication like receptors, just so many different functions of the body. So proteins are made of the monomers called amino acids. There are 20 amino acids found within the body. And the actual proteins themselves, though, can be thousands and thousands, tens of thousands of amino acids long. So even though there's only 20 different amino acids, the combination and the way that they're arranged in these long, long chains can really determine the overall shape of the protein and the function of the protein. So when proteins are, let's talk about amino acids first. So a single amino acid has a central carbon. And around that central carbon then it has several different components. I'll point out this. So here's our central carbon, and then we'll have a hydrogen attached with one bond, and then we'll have a carboxyl group. We talked about functional groups. So here's a carboxyl group on one end, and then on the other end we have the amino group. So carboxyl is an acid, and here is our amino group. So that's where we get the term amino acid. And then the important part is, so all amino acids have the carboxyl group, an amino group, then and a hydrogen and a central carbon, but then this R group or variant group right down here is what really, there's 20 different R groups, and so those make up the 20 different amino acids. And so we call that the functional group or the variant group or the R group that's attached to the central carbon. Here are the different 20 amino acids. You've probably heard of a lot of these. I'm not going to ask that you memorize all 20 amino acids, but I would like you to take a look at these different functional groups here. And we have three major categories. There's the non-polar category, and we've talked about polar and non-polar before. So non-polar amino acids have equal distribution of electrons, and so they're going to be more of the fats, and they're not going to interact well with water. Then we have our polar amino acids here, and then we have our electrically charged group down here. So, and that includes acids and bases. So those are some major categories. I'm just going to point out ones you've probably heard of, things like tryptophan or tyrosine or other big ones. Well, they're all kind of all big. Glycine, valine, leucine. I'm not sure which ones you've particularly heard of, but maybe all of them. But here are the 20 major 20 amino acids in their major groupings. So these amino acids are bound to each other to form the polymers of proteins, and this requires a dehydration synthesis or condensation between two adjoining amino acids. And in this case, the amine group of one amino acid will join with the carboxyl group of the next amino acid. So there is sort of a front and back type of orientation. And imagine this kind of like a train. When the cars are all attached to each other, there's a specific linking that needs to occur in a specific fashion. And so this binding of one amino acid to another amino acid forms what we call a peptide bond. And this peptide bond is very, very strong, and it holds these two amino acids together. And then you keep stringing more and more amino acids together, and now you have what we call a polypeptide. Poly, many, peptide, many bonds. So we have a longer string of amino acids. So a little peptide, we just call it peptide, maybe a few amino acids together. So less than 50, for example, are just called little peptides. And then at the end of this train, we kind of have the engine and the caboose. So there is still what we call an amine group on the first amino acid, and we call this the N terminal. And then there's a carboxyl group on the last amino acid, and we call this the C terminal, C for carboxyl, N because there's nitrogen in amine. So those are, and so there is sort of a front and a back, and that can also then be joined with other molecules as well. Here's an example or picture of a polypeptide, and we have, you can see in this picture, here's our amine group and our N terminal, and here's our carboxyl group or carboxylic acid in our C terminal. And here are, and R is just signifying that these are functional groups, and there could be any variety of the amino acids here. So we have, this would be one amino acid here, and this would be another amino acid here, and all the purple boxes are indicating the peptide bonds between each amino acid. So you can see the different functional groups as indicated by the R's. So this is a string of amino acids forming a peptide, or if it's bigger, it's called a polypeptide. So if they're super short, they're called oligopeptides. That's like 3 to 20 amino acids. 21 to 199 we call peptides or polypeptides. And then anything bigger than 200 or more, which are the really big proteins in our body, we just call them proteins. There can also be another type of protein, still a protein, but they can have carbohydrates sort of attached on the side, and I'll just name it like this. Some maybe carbohydrates kind of coming off of the protein backbone, and we call those glycoproteins. So protein structure is incredibly important in function, and I cannot emphasize this enough. And you'll see some different mutations in proteins as we go throughout the semester, and certainly as you go further in depth in pathophysiology. But protein structure determines its function. So not only do proteins have a very specific order of amino acids, so there's the way that the amino acids are arranged, so the order of the cars of the train are really, really important. And this is determined from the DNA. And the DNA codes for specific amino acids to be strung together in a particular order. So the way that the amino acids are strung together is called the primary structure. So maybe you'd have an alanine and a valine and a tryptophan, and it's just a very specific order. It's called the primary structure. Sort of think of multicolored beads on a string. And then the way that these amino acids are bound together causes them to sort of loop and start looping or folding in particular fashions, and this is called the secondary structure. They kind of make two primary structures. They either twist up or twirl up, kind of like curly hair or a spiral, and this is called a helix, an alpha helix, or they can kind of kink and bend, and think about paper being folded back and forth like a paper fan or something, and these are called beta pleated sheets. And then those structures, so there's probably a bunch of beta pleated sheets and a bunch of alpha helices in one big protein, and then all of those will start flopping in on each other, kind of like a ball of string, being just kind of crumpled in, and you got like this glob now of string, and that is the tertiary structure. So this is really the structure that determines function. And even if the primary structure, all the amino acids are in the right place, and the alpha helices and the beta pleated sheets are fine, if something comes in and interrupts the way that this totally folds in like a three-dimensional structure, maybe it's twisted or bent or folded, kind of kinked a little bit differently, it could cause maybe a binding pocket to be not available. It could cause some sort of reactive group to be facing outward. It could really change the function of the protein. So this tertiary folding is incredibly important, and oftentimes the tertiary folding is affected by some changes in the secondary or primary structures. But really these folding of the proteins is incredibly important in its function. Some things that could change the tertiary folding could be enzymes. Temperature can cause proteins to sort of unfold and go back to just a linear protein, and then it has no function. Different pHs If environment becomes more acidic or more basic, that can affect the tertiary structure and cause the protein to lose its function. So there's a lot of different environments and things that can happen to cause this protein to stop working. And sometimes that's used as part of how we turn on and turn off proteins, is maybe another protein will bind to it and cause it to change shape. Maybe there'll be some chemical gradient that causes it to change shape, and all of these an enzyme that causes it to change shape. So that's how we turn different things on and off. So that's just so important in basic biology to understand. This is how we activate a receptor. This is how we initiate a signaling cascade or propagate the signaling cascade. This is how hormones function and work, and how they stop working, and how they're degraded, and half-lives and efficacy. Just everything is based upon protein structure. There's also a quaternary structure, and this isn't with all proteins, but sometimes proteins have multiple subunits that come together. So there'll be 4 three-dimensional structures, for example, that will come together and form what we know as hemoglobin. And there's globin molecules, 2 alpha and 2 beta. So there's almost like 4 mini proteins that come together to actually create the fully functional final protein. And those are proteins that have a final quaternary structure, but not all proteins have that. Here's the primary structure. It's a linear sequence of amino acids that are joined together by the peptide bonds. Here is an example of secondary structure. So here we have the alpha helices, which sort of form these twisted, twirly loops or spirals, and then here we have beta pleated sheets, which are more kinked type of structures. And depending on the arrangement of the primary structure of the protein will what amino acids are next to each other will cause them to twirl into these alpha helices or kink into these pleated sheets, and it's not. And then there's also parts of the protein that don't form these structures. But alpha helices tend to kind of, think of a coil or a slinky, kind of tends to stretch a little bit more, so we end up with more of these structures in more of elastic fibers. Beta pleated sheets sort of allow flexibility for more like big globular proteins like enzymes and things like that. So they do a little bit different, but these are usually found in both of them are usually found in every protein. So here we have the tertiary structure, and here you can really see that there are, here's some alpha helices, here's some pleated sheets. So there's multiple helices and multiple pleated sheets found within this this is your 3-dimensional structure. And here you can, for example, this one's with a lot of alpha helices, and these will give a lot of more like elastin and proteins like that that are supposed to be really stretchy have a lot more alpha helices. And then here's an example of quaternary structure where you have, and here again this is hemoglobin here, where you've got 2 beta and 2 alpha globulin, so that's our quaternary structure there. This is collagen here, where you see 3 strands of proteins that are twisted together to create a strong protein. So I'll just kind of test your knowledge here. You can see if you can answer this question to see if you're grasping the concept. This is a pretty simple question, so just kind of making sure we're all on the same page. Okay, next we're going to talk about nucleic acids and their individual monomers and the final part. Nucleic acids are, there's 2 major types of nucleic acid, and these are important for either storing genetic information or transferring genetic information. In other organisms, RNA, as one type of nucleic acid, can be the primary molecule that stores genetic information, but in humans, DNA is what stores our genetic information, and then for us, RNA is useful in transferring the information so that it can become, the information from the DNA can become a protein, or give information on how to make that protein. So both of these types of nucleic acids, RNA and DNA, are composed of smaller monomers called nucleic acids, or nucleotide monomers. And these are bonded to each other not through a peptide bond like proteins, but rather through phosphodiester bonds. And so DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And we'll go through the major components of these. So the nucleotide monomers have three major parts. Just like we talked about an amino acid and the central carbon and the components of that, we're going to kind of go through some really basic parts of a nucleotide. And here we have a sugar right here. So this sugar is a pentose type sugar. It's a 5-carbon sugar, and that's what the little structure here is. There's little carbons at each of these joining angles here. And then we have a hydroxyl group here and other things like that. So this sugar is important because it can be a ribose or it could be a deoxyribose. So if it has an oxygen here, it's an RNA at this spot. If it has a hydroxyl group, so that's ribose. If it does not have an oxygen or a hydroxyl group, so it's deoxygenated, no oxygen, then we call it deoxyribonucleic acid. So that is the sugar, this ribose, or a sort of pentose type sugar here. Then we have a phosphate group that's attached here, which is attached to the sugar. And then also over here, importantly, we have the nitrogenous base. And this can really vary. So this is sort of like the functional groups in the amino acids. So in all DNA, the sugar is the same. It's missing that hydroxyl group, but it still has the phosphate group, and then we have these deoxyribose groups. But then there can be different nitrogenous bases. And in RNA, all the sugars are the same. They're just ribose sugars with that extra hydroxyl group right here, or in this spot. And then they also have the phosphate group, and then they can have different nitrogenous bases. So that's really our what makes the different types, and I'll talk about the major types of nucleotides. So there are 5 major types of nitrogenous bases, and when they're attached to the sugar, then that creates your different nucleotides. So there are 3 pyrimidines. There's 2 classes, pyrimidines and purines, and there are 3 pyrimidines and 2 purines. So the pyrimidines include cytosine. We abbreviate that as a C, uracil, which is a U, sorry about the mouse here, and then thymine, which is a T. So these are all pyrimidines. They're single ring bases. The other class are called purines, and they're double ring. And I have this down here in this image down here. We have our purines and our pyrimidines. So the purines are adenine or an A, or guanine and is a G. So those are our 5 types. Now in DNA there's only 4, and in RNA there's only 4, and really it's these 2 that kind of swap out for each other. So this one is in DNA. That's funny. I look like a kindergarten handwriting here. And this one is in RNA. And then the other 3 are the same. So that's the only really difference there. Also, while we're looking at these pictures, we always have pairing of bases together. So DNA is double-stranded. So here's our 2 strands. And so there'll be one that is on this side, and it will always have a purine pairing with a pyrimidine. So a single ring structure pairing with a double ring structure from that side. And it doesn't matter which side of the DNA they're on, but always have a double ring pairing with a single ring. Let's talk a little bit about DNA and its structure. So DNA is double-stranded. It's again a nucleic acid. It's located inside the nucleus. It's very, very long and crazy stringy. When cells divide, instead of trying to separate out all this DNA and get it all tangled up, the DNA actually condenses into very, very, very coiled structures, and that's what we call chromosomes. Just highly condensed, coiled up DNA. But then when the cells are not dividing, the DNA sort of unwinds, and certain parts are more unwound than others, and that allows molecules to come in and bind to the DNA and start the process of transcription and translation. So again, there's always a deoxyribose sugar, so we're missing the hydroxyl group. There's always a phosphate, and then there's one of the 4 nitrogenous bases. And again, here they are highlighted for you: adenine, guanine, cytosine, and thymine, but no uracil in DNA. The 2 strands of DNA are held together by hydrogen bonds, and then the complementary bases bond to each other, again, a purine with a pyrimidine, or a pyrimidine with a purine. And the way that these always form is adenine is always pairs with thymine, and guanine always pairs with cytosine. So AT and GC. We never mix them up. It's never GT or AC or CT or AG. It's always AT and GC. So I always think of this says AT, and then the 2 curvy letters. So a C has sort of a curve and a G has a curve. So that helps to remember which ones go together. Here is a picture of our double-stranded helix, which is our DNA, and the pairing of the nucleotides together. So here you can see C and G going together, A and T. And really, what makes all the DNA different or have a message is the order of these. So whether there's 6 A's and there's only 4 different letters or nucleotides in DNA, but you can arrange them in millions of different orders, and the order could be 10 nucleotides long, which would be a gene, or it could be thousands long. So genes are segments of the DNA that code for something specific, and it's really just the order of A, T, C, and G that determine what a gene has to say or what instructions it holds. It's really kind of amazing. So we have RNA, or ribonucleic acid, and again, this is just that sugar has a hydroxyl group on it, but then we again have a phosphate and also a nitrogenous base. And here, adenine, guanine, and cytosine are the same, but we have the uracil is different, so there's no thymine here. And the same pairing applies when RNA binds to the DNA to kind of take the message from the DNA and it's going to bring it somewhere else. We still have the same type of pairing. We still have G and C pairing together, and we have adenine pairing with thymine, but then uracil pairs with the adenine. So it's just a little bit different, just swapping out the thymine for the uracil in RNA. There are 3 kinds of RNA. There's what we call messenger RNA, and this is probably the most well-known RNA in the human body. And so this is the RNA that will bind to the DNA and basically make a template of whatever nucleotides were on the DNA. The RNA just matches it up. It's actually would be inverse of it, but it takes the order, that really important order of nucleotides, and it kind of copies it. And then it will take it out of the nucleus to a ribosome, where that order will be used as a blueprint for making a polypeptide protein, or a long chain of amino acids. There are these amino acids that are also attached to a type of RNA called transfer RNA. So each of the 20 amino acids have a little transfer RNA that they're attached to, and that transfer RNA comes in and attaches to a ribosome. And here's kind of a picture of that. So here's the transfer RNA attached to an amino acid right here, and it will come in and bind in the ribosome and sort of drop off its amino acid, which will get which will have a peptide bond to the next amino acid, and then the transfer RNA will go away. And so this is sort of just bringing in each hauling an amino acid and dropping it off, and then the transfer RNA leaves. And then we also have ribosomal RNA, and this is used to make the actual ribosome mechanisms. So I want to talk a little bit about a nucleotide that's really important that isn't actually DNA or RNA, but it's adenosine triphosphate, or ATP. And this has adenosine attached to it, so it does have a nucleic acid or a nucleotide. So it has a nitrogenous base, our adenosine right here. It has a ribose sugar, and then it has three phosphates. So triphosphate and adenosine triphosphate, ATP. And I'm sure you've heard of ATP before, but that's where it gets its name. It has an adenosine, a ribose sugar, and then three phosphates. And really, these three phosphates are important for energy. And so there's covalent bonds between these phosphate groups. So there's some energy in this bond right here, but there is a lot of energy in this last bond between phosphate 2 and 3. And when that bond is broken, it releases a whole bunch of energy. So ATP can be used to generate, or used as sort of energy currency by breaking that last little phosphate off, and then it becomes a DP, di meaning two, so adenosine diphosphate, meaning there's only two phosphates, and that you could still use it to break that other phosphate bond and get some energy, but it's definitely not as good as the triphosphate. So a DP usually then goes somewhere and gets that another phosphate attached to it, and then it can be TP again. So that's the conversion of ATP to ADP, and that's kind of how we generate or store energy. And so it's really that third phosphate bond that has so much energy in it. So here's just a little overview of the difference between DNA and RNA, whether it's their actual structure, the nucleotides that are present, where they're found. So DNA is only in the nucleus, for example, and RNA goes into the nucleus like messenger RNA, but then also goes out into the cytosol, and kind of what they do, and then the difference between their sugars as well. So it's just kind of a comparison between the two for you. It's a little bit of a review. Here is a sort of easy question. So this is just testing your understanding to see if you are on track as far as which nucleotides in DNA pair together and which one is incorrect. So you'll see a lot of questions like this on quizzes and exams, so make sure that you read it carefully. And usually what I do is I look for ones that I know are correct, and I will put like a true next to it or a false next to it, and true, false, and then I have kind of all the things. So then I go just double check the question. I'm like, okay, we're looking for incorrect or false, and so then you can select the correct answers. So make sure that you're reading questions very carefully. When I ask you which of the following is correct, which of the following is incorrect, or which of the following is true, which of the following is not true. So make sure that you're reading those very carefully, and then first thing to do is just cross off ones. I don't even cross them off. I just write true and false next to them, so that I can really, and read it very carefully, so I can really tell which is the correct answer. Next, we'll be talking about carbohydrates or sugars, complex or simple sugars. This is just a little cartoon of a carbohydrate in the dressing room talking to his friend a lipid. So carbohydrates, another biomolecule, contain carbon, of course, and hydrogen, like all biomolecules, and also contain oxygen in their makeup as well, in a greater degree than we see in proteins or nucleic acids. And they have a very specific ratio. You don't have to get caught up in the chemical structure. I just kind of wanted to show you an example of a carbohydrate. So there's a couple different stages of complexity when we're talking about monomers and polymers of carbohydrates. So the monomers of carbohydrates are actually called monosaccharides. Saccharide means sugar. So monosaccharide means one sugar. And there's different kinds of saccharides. And some examples are glucose, fructose, galactose. these can get coupled together in different ways, maybe two glucose or a glucose and a fructose. So they get bound together as a doublet, two. And so we call this a disaccharide, di meaning two sugars. And then when they're coupled together as two, they're given new names, of course, right? Need to make it more complicated than it was before. But here we have, for example, a glucose and a fructose, and those coupled together are called sucrose. A galactose and a glucose here are called lactose. Two glucose together are called maltose, right? So that's our more complicated naming, but you've heard of lactose, you've probably heard of sucrose, you've absolutely heard of glucose before. So these are sort of the two lower levels of complexity of sugars, a monosaccharide and a disaccharide. And they are condensed, or a dehydration synthesis, where we were removing, again, a water and then using this oxygen to covalently bind these molecules together. So after a monosaccharide and a disaccharide, then we have just more complex sugars. So we call these polysaccharides. And these are all linked by covalent bonds in the same way that disaccharides are linked together, and they can get super complex. So glycogen is a very complex polysaccharide. Starches, cellulose, they're just, and here's a little picture. All these little rings are supposed to be individual monosaccharides, and you just see that they're highly branched and linked together. So this is starch, for example, and it contains a bunch of glucose molecules. So I thought it's worth giving glucose its own little slide, so that because it's such an important molecule, it's our #1 sugar that our body prefers to use as a source of energy. It's a basic 6-carbon carbohydrate. Here it is. You don't have to know the chemical structure, but I'm just putting it out there to show you what it looks like. It's the most common monosaccharide, and again, the primary nutrient for our cells and what they mostly prefer. And it's also really important to maintain glucose levels within the blood, and so this can play a huge role in diseases like diabetes. And so insulin really is important in controlling glucose levels within the blood. Glucose if there's too much glucose in the blood, it can clog up a bunch of things and cause all sorts of problems. So really maintaining appropriate glucose levels obviously plays an important role in human health and disease and maintaining homeostasis within the body. So if there's extra glucose in the blood, and here's an example, glucose 6-phosphate is the actual type we use, that can actually get converted into glycogen, which we store away in the liver and in the muscles. So if we have extra glucose in the blood, like after we eat a meal, we store it away as glycogen through a process using insulin. And then between meals, when our glucose levels of the blood drop, glycogen is then converted back into glucose in the blood so we can maintain our glucose levels. And these processes are called glycogenesis. Genesis means the creation of, kind of like Genesis in the Bible, the creation of the world. So the creation of glycogen, glycogenesis. And then lysis means to break. So glycogenolysis is the breakdown of glycogen. So those are the two processes that sort of control those glucose levels within the blood. Lastly, we are talking about lipids or fats. And these have very special properties that give them unique things that they can do. And also there's unique subunits, the monomers, that make up these larger fats or lipids. So lipids are mainly hydrophobic, meaning that they don't interact with water, and we kind of all know that oil and water don't mix well. And the properties that make them hydrophobic are the fact that they're mostly nonpolar. And nonpolar water is polar, and like dissolves like. So polar molecules dissolve in polar water. Nonpolar molecules do not dissolve in polar water. These again are made we have a carbon backbone and hydrogen atoms around them, and these hydrogen atoms are usually uniformly distributed around them, so there's not these amine groups or carboxyl groups that are sticking out, or functional groups that are sticking out of these lipids. Otherwise they would end up having a polar part of them, and a few of them do, but for the most part they have a pretty non-polar sort of structure. And these lipids include things like fatty acids. Now fatty acids do have an acid component to them, so they can be somewhat reactive. Glycerides like triglycerides, which include fatty acids in them, steroids, phospholipids, glycolipids, eicosanoids. These are all types of lipids or fats found in our body. So the most important or biggest type of lipid that's in our body are what we call triglycerides. Tri means 3, and then we have a glycerol molecule that's part of this. So a triglyceride is our most common form of lipid in our body, and it's used for storage. So this is what creates the fat on the hips and the butt and the stomach and our legs, and I could go on about the different places that fat can be stored. And it's a really high and dense energy, and it takes up a small amount of space. It's actually a really efficient way for our body to store energy as these triglycerides, rather than just storing a bunch of glycogen, which actually are pretty large molecules and take up a lot of space. So these are high energy, low space molecules. They also provide, like, in the hypodermis, they're providing cushion and insulation in our body as well. So the structure of a triglyceride has a glycerol molecule here, and that's where the glyceride comes from. Tri is 3 fatty acids, 1, 2, 3 fatty acids that are attached to this glycerol. And so here you can see it, and usually one of them is sort of kind of kinked right here on the leg right there. So this is a triglyceride, and this can be broken down into glycerol and 3 fatty acids. Here's kind of a more simple structure of this glycerol and 3 fatty acids. And these fatty acids can get kind of broken down into individual fatty acids, which then can be absorbed a little bit easier in the digestive tract. But that's kind of in general what they are. They're formed by dehydration synthesis, so removing that water or condensation, and forming these molecules together. And they're broken apart again by hydrolysis, so the type of bonding and breaking that we talked about with most of the monomers and polymers. So that is our kind of big fat, our triglycerides. So this would be sort of our polymer, and our monomers would be fatty acids and glycerol. So individual fatty acids are carbon chains that are surrounded by hydrogens, long carbon chains like this, and they're usually uniformly surrounded by all these hydrogens at each carbon. And then we do have a carboxyl group, or carboxylic acid, at one end. So that's what gives it the fatty acid. And this little group right here does make it a little reactive with other groups, and where it gets its name from. It's relatively nonpolar. Most of the molecule is nonpolar except for that little end right here. So the fatty acids can be what we call saturated with hydrogen. So every carbon has a hydrogen attached to it. So this is completely saturated. There's not a missing hydrogen. If there is a missing hydrogen, what can happen is a double bond can form between 2 carbons, because there's sort of this, if you pull off a hydrogen, you're losing an electron, and those valence shells do not like to be missing something, and so a double bond will actually form, and it causes this fatty acid to sort of bend and kink in a certain way. So we call one a saturated fat and the other one unsaturated, or if it's unsaturated in several places, a polyunsaturated fat. So there's 1, 2, or more double bonds formed there, and there's different health implications in saturated and unsaturated fats. So just as a little test here, this is lauric acid. Is it an unsaturated fatty acid? So just take a look at the structure. It is and you can see no double bonds here. If you'd see a double bond, there'd be 2 lines. So we do not see any double bonds. So this is a saturated fatty acid. So the correct answer is no. Steroids are another important type of lipid. They are derived from cholesterol. So cholesterol is a lipid, major component in all of our steroid hormones within our body. And here's our parent cholesterol molecule. So cholesterol is a good thing, but too much cholesterol can be bad for you. But cholesterol is what we need to make a bunch of hormones, which are really important. So here's our parent cholesterol made of these 4 rings. And then cholesterol gets broken down, rearranged, kind of things are moved around to make a bunch of different steroid hormones. And that includes things like testosterone and the estrogens like estradiol and estrone. Also hormones like corticosteroids, vitamin D. So there's really important implications in these type of lipids. Also, because of their nonpolar nature, they are able to move through a phospholipid bilayer of the cell membrane without needing to bind to a receptor. So they can directly enter cells without needing any sort of help to do that. So that has a lot of implications for target cells and efficacy and potency of hormones in different cells. Phospholipids are incredibly important lipid found in our body. These little guys make up the phospholipid bilayer of every single cell of your body. So phospholipids have a phosphate group that attaches to sort of a non-lipid group here at the top. And so this is called kind of the head of the phospholipid. And then here's a glycerol molecule like we saw with our triglycerides, but then we just have 2 fatty acids, one kinked, one not kinked, that come off. So this is our structure of a phospholipid. And these guys are arranged with all of the heads, which actually are hydrophilic, that lay like water. They all arrange together, so these are polar, and then their little tails, which are hydrophobic, this is the lipid component, the non-polar, arrange and face the tails of another phospholipid, like this. And so the hydrophobic tails that face each other kind of create this middle section here that's a hydrophobic section. You can see that down here. So this inner area in here is hydrophobic. It does not like water. And that's why these tails sort of cling to each other, like to come by like. So we have hydrophobic inner core, and then out here is water. And in here is water. This is the inside of the cell, and this is the outside of the cell. And because it's facing water, these are polar heads. They like water, so they face the water on the inside and outside of the cell. So it creates this, what we call a 2-layer, a bilayer, made of these phospholipids. And so the structure of the phospholipid is actually really important in how they arrange in the bilayer, and it's the reason that this bilayer exists the way it does is because of these chemical components and the structure of these phospholipids. Last type of lipid I just want to briefly mention are eicosanoids, and these are derived from different fatty acids and arachidonic acids, and these include chemicals like leukotrienes, which are important in the immune system, prostaglandins, which are short little fatty acid chains that are important for a little local hormonal regulation, and thromboxanes, which are really important in the clotting cascade and breaking up clots and preventing clots and all of that stuff. So this is just an extra group of lipids that are out there that include some of these other peripheral things that you may or may not have heard of before.